Okay, the next unit, which is blood. So chapter 19, blood. The blood is type of connective tissue. If you still remember that from 233, when we talk about the different types of tissues, connective tissue, muscular tissue, neural tissue, this is the connective tissue. And it's basically fluid matrix, which is watery fluid, okay, that contain other materials dissolved in, and we'll talk about this, and cells. So we're talking about fluid and cells in that fluid. Generally speaking, the function of the blood is transport dissolved substances like oxygen, carbon dioxide, right? Oxygen will be transported in the blood to go to the tissues. Carbon dioxide will be transported in the blood to move away from the tissues and goes to the lung. Regulation of pH and ions, we will talk about this. It is actually controlling the pH by acting as a buffer and so on. We will talk about this. Restriction of fluid loss at injury. If you have an injury, your blood will coagulate and will stop to save itself so you don't lose it anymore. Defense mechanism, and this is basically all the details about white blood cells, and this part here is about the platelets. And stabilization of the body temperature, we talked about this before, and when it's cold, you see your blood vessels in the skin are getting constricted and you look really pale. While if it is getting warmer or hot, the blood vessels dilate and you see your face flushing or becoming more pink or red. So these are the general functions of the blood. So the whole blood consists of two parts. The plasma, which is the fluid part. So the fluid part is called plasma. And formed elements. And these are the cells and solids. Okay, so we have two parts. Plasma and formed elements. Plasma is the fluid. Formed elements is others, like solids and cells. What are the cells? Red blood cells, white blood cells, platelets. The plasma itself is just water and plasma proteins dissolved in that water, and this is the main thing, plus other solutes that we will talk about. There is always an exchange between the plasma and the interstitial fluid. Okay, and I think we covered that before. Do you remember from the heart, the blood is pumped out and go to the aorta, from the aorta to the large arteries, to the small arteries, to the arterioles, then to the capillaries. The capillaries are the smallest blood vessels in the body, right? We know that. At the capillaries, because they have pores and they are one cell layer only, endothelial cells, so things can move out from the capillaries going to what? See if you understand it or not. If this is oxygen in the plasma, inside the capillaries, it will leave the capillaries and go to another fluid. What is that fluid? Outside of the capillaries. Where do you go if you leave the capillaries? Don't remember? Okay. I don't know if they have one of these markers or not. Okay. Oh, thanks. Thank you. So these are the capillaries. Okay. This is the arterioles, and these are the venules. Like the arterioles are coming from the small arteries, and the small arteries are coming from the large arteries. Right? Are we following? from the aorta to the large arteries, small arteries, arterioles, and then the capillaries. And what is this? Not the veins, the small veins. What is it called? Venules. So these are arterioles, and these are venules. And then small veins, large veins, right? And what's the last thing before the heart? Vena cava, superior and inferior vena cava. So what's happening here is the blood will come like this, and it goes to the capillaries, and the capillaries obviously have a lot of pores. Okay, so oxygen will leave and go in between the cells. So these are the cells. And what do you call this in between the cells? There is a fluid here. What do you call it? Huh? What is it? Cytoplasm. No, cytoplasm is inside the cells. What is outside of the cells in between? Interstitial fluid or extracellular fluid. Interstitial fluid. What is the interstitial fluid? The cells are not all in contact. There are spaces that have fluid in between the cells. Okay? That was the very, very beginning of 233. So obviously, just like everything else. Okay. I'll keep on blaming you until the end of the semester. Okay, so oxygen, for example, leaves the capillaries, goes out. What's out? It's the extracellular fluid or the interstitial fluid, and then it will go to the cells. The cells need oxygen, right? It will take it, use it, and it will produce carbon dioxide. Carbon dioxide will leave the cells, go to the interstitial fluid, go back to the capillaries, and go back to the heart. The heart is in between those two. Okay? So this is the exchange happening across the capillaries. Good things leave the capillaries, go to the cells, like oxygen, nutrients. They leave the capillary bed and go to interstitial fluid. Then the cells will take it. And when the cells take nutrients and oxygen, they're going to burn the nutrients with oxygen. You produce the energy, so you got what you need. And then you release what you don't need, which is waste products and carbon dioxide, right? Isn't that what the cell doing? The cell all the time. You take nutrients, you take oxygen. You use the oxygen to burn the nutrients and get energy. That's how you do it. At the end, you release the bad stuff. What's the bad stuff? Carbon dioxide and waste products. So what goes out is the good stuff. Go to the cell. What goes back is the bad stuff. Carbon dioxide and waste products, and they go back to the venous system. Okay, so now the cells, we have different types, 3 different types of cells. We have red blood cells, white blood cells, and platelets. Red blood cells have another name, which is erythrocyte. Erythro means red, cytes mean cells, and leukocytes, what's leuko means? Huh? White, leukocytes, the white cells, and platelets. And the general function, there are other functions, but transport of oxygen and other things. White blood cells, immunity, it's responsible for defense, body defense. And the platelets is basically blood clot, to stop you from bleeding. This is the general function. Hemopoiesis is the process of producing formed elements. So hemopoiesis, what's poiesis or poiesis? Poietic. We mentioned that several times. What's poiesis? Producing. And he means blood. It doesn't mean red cells. It means the whole, all blood cells. So hemopoiesis or hemopoiesis is the production of the blood cells in general. Whether we're talking about red cells or white cells or anything else. Where is it produced at, or what's the part of the body that produces it? Myeloid and lymphoid stem cells. And anytime you see stem cells, these know that this is the origin of everything. Everything have a stem cell from which everything else will be generated. Okay? Fractionation is when you separate the blood into 2 fractions. What are the two fractions of the blood? What are the two parts of the blood? Plasma and formed elements, which is the cells and particles. Generally speaking, the blood is 38. Viscosity is high. And what's the pH of the blood that you need to remember? I told you of before. What's the range? This is a number that you have to remember. 7.35 to 7.45, or 7.4 plus or minus 0.5. But you have to say a range. Okay? Because 7.35 is okay too. 7.37 is fine. 7.42 is fine. Anything in the range is fine. Okay? So 38, what's a normal body temperature? 98.6 degrees. In Celsius. What's the normal? Degree centigrade. What is it? 37? Yes, 37. So this is a little bit warmer, right? If you look at both of them, it's the body is 37 or 98.6, right? So it's a little bit higher. Yes, the blood is usually a little bit warmer than the body, and this is how the blood is going to regulate your body temperature, because it's warmer. So if it is really warm, it's warmer, so it takes the heat and goes out to the skin, so you lose the heat. If it is cold, the blood vessels constrict to keep the blood warmer than the body, so it's warming the body. Did you get the idea from this? The blood volume is about 7% of the body, but these numbers is something that you need to remember. 5, 6, 4, 5. 5, 6 liters. If I counted all your blood as a male, if I get all the blood and measure it, it's 5 to 6. Females are 4 to 5. Here is the rule of the thumb. In blood, specifically, anything in males is higher than females. Period. Anything. Okay? Anytime we talk about anything, and we have two things, the higher is for males and the lower is for females. Not only the volume, not just 5, 6, 4 to 5. So it's 1 liter less, right? That will apply for everything. All the numbers. Okay? So plasma, which is the fluid part, 92% of it is water. So it's watery fluid, watery liquid, right? It's liquid, watery. Plus 7% plasma protein, which is the major part after the water, the main thing. So number one is water, 92%, huge. This is the vast majority. Plus, or the major part, major volume, 7% plasma proteins, and everything else is 1% only. Okay, plasma proteins. This slide is very important. Okay, it contains a lot of information that's important. Plasma proteins, 7%. What are the plasma proteins? Albumin, globulin, fibrinogen, and regulatory proteins. Okay, regulatory proteins are just enzymes and proenzymes and hormones. So we need to know that these can come from the plasma proteins. But the function of each one of those is important to remember. So why do we said that the plasma consists of water plus 7% plasma proteins and 1% others. Just small amounts of everything else. 7% plasma proteins. What are these plasma proteins inside our body, and what's the function of each one of those? This is important. All right? It doesn't mean that anything else is not important, but this is the core. You have to know it. So what do we have? We have albumin, globulin, and fibrinogen. What's the albumin for? Osmotic pressure. This is what make our osmotic pressure. Do you remember the osmotic pressure? Or just like everything else in 233?
Everything else.
Just like everything else? Okay. Do you remember the osmosis? The movement of water? Does the water move from more concentrated solution to less concentrated water? Or from less concentrated to more concentrated? What do you think? Water.
High to low.
It's low to high. I understand that if you think about it, of course it's high to low, right? If I have 5 here and 2 here, 5 go to the 2. Yes, that's if I'm talking about the solute. I'm not talking about the fluid, I'm talking about the water. The water is unique. The water follow osmotic pressure. If I have 2 compartments, okay? I have 2 compartments like this. And understand this, because it will help us in other things. If this is a container and I put something semi-permeable membrane in between here, and this is 1 liter and this is 1 liter of water, right? And I put 10 grams of salt here and 5 grams of salt here. This is 1 and this is 2. Which one is more concentrated? 1 or 2? 1. It's 10 in liter and this is 5 in liter. The water will actually move from this to this. Got it? From less concentrated to more concentrated. But you're saying salt. Yeah, salt make osmotic pressure. Okay? Albumin, but albumin is more important. So how about albumin? I put 10 units of albumin here and 5 units of albumin here. The water, I'm not talking about the albumin. I know the first thing came to your mind is you have 10 here and 5 here, so 10 go to the 5. Yeah, this is for albumin. Did I say albumin? Did I say salt? If I say salt, yes, you say from 10 to 5. But this is the opposite. Water moved from less concentrated to more concentrated. Don't ever forget this. Water follow osmosis. What's osmosis? Water move from less concentrated to more concentrated solution if you have a semi-permeable membrane. What's the semi-permeable membrane? It's a membrane that allows water only to go through. So why the albumin? Shouldn't the albumin move the other way around? Yes, but can the albumin actually go through the semi-permeable membrane? No. This is the plasma protein. It's so huge. It's not as small as the water. It's large. So it cannot go through. And this is what we mean by semi-permeable. It's half permeable. So it can allow small things to go through. But plasma proteins, that's big. Okay? So this is the most important thing to remember about this. Plus, it transports things. Transporter. Like it carry lipids, for example. It carry the lipids. It can carry other things. So 2 things to know about the albumin. Osmotic pressure, carrier. Osmotic pressure and carrier. Albumin. Globulin, the most important thing to remember is antibodies. Immune system. The antibodies are actually globulins. So if I ask what's the nature of antibodies, they are globulins. What's a globulin? Plasma protein. It's part of the blood, right? So this is the most important thing to remember. What else? Transport. Also, yes, both of them transport. Albumin transport, globulin transport. What's the unique about albumin? Osmotic pressure. What's the unique about globulin? That only this do it? Immune system or antibodies. How about fibrinogen? Fibrinogen we will talk about it later, but fibrinogen. What gen means? Makes or generate? It means generate. Or makes is the same thing. So fibrinogen. It's going to generate fibrin. So this is the precursor of fibrin. Okay, so fibrinogen, when you activate it, you remove the gen and it will become fibrin. What's the fibrin? Fiber. What's the fiber? The fibers, if you have a wound or something, these fibers will go and lay itself on the wound so that you don't bleed anymore. So this is the basic. The fibers go and lay down like this, and platelets will come, interact with these fibers. That's how we stop bleeding. Okay? So if this is a wound like this, okay, how to close it? Fibers will go like this, which is the fibrin, and then the platelets will go like this to temporarily close the wound, okay, or the cut in the blood vessels, right? So this is plasma. Okay, so you said these are the cells like this, right? And what did we call this? What is this? What do you call it? Interstitial fluid? Interstitial fluid, yes. Is this interstitial fluid is inside the cells or outside?
Outside.
Outside. How about the plasma? No. The plasma, the water here, is it inside or outside of the cells?
Outside.
It's still outside, right? So both of them are called extracellular. Means both of them are not inside the cell. So how about the water inside the cells? You call it? It is part of the cytoplasm, but what do you call it? This is called extracellular.
Intracellular.
Intracellular, yes. Intracellular. So the water is, or the fluid is, fluid in general, not just water, is either within the cells or outside of the cells. If it is within the cells, you call it intracellular. If it is outside the cells, you call it extracellular. To be more specific, it's outside of the cell, but where? Inside the capillaries, that would be intracapillary, okay? Or meaning in the blood vessels. Or is it in between the cells, you call it interstitial fluid. Did you get this? Fluid, inside or outside? Inside intracellular, outside extracellular. Outside where? In the capillaries, this is the blood. It's a plasma, I mean. If it is in between the cells, you call it the interstitial fluid. And again, it's water, solutes, which what we talked about, plasma proteins and ions. So now what's the difference between the plasma and the interstitial fluid? Okay, I'm going to ask you and try to figure it out. This fluid inside the capillaries is different, a little bit different than the fluid in between the cells. So the plasma fluid, the plasma, is a little bit different than the interstitial fluid. Okay? If I told you, excuse me, thank you. If I told you one of those have more oxygen, which one? In the capillaries, which is the plasma, or the interstitial fluid?
When? Before or after?
Before, like here.
Capillaries.
The capillaries have more, so it will go out. Which one have more carbon dioxide? Here, so CO2 is more here, right? So it's less in the capillaries, okay? Actually, it's always, interstitial fluid is always higher in carbon dioxide and lower in oxygen, and the capillaries are the opposite. Higher in oxygen and lower in carbon dioxide. So this is the first difference. The amount of carbon dioxide and oxygen. Otherwise, just think about it logically. Otherwise, if the oxygen in the capillaries was not more than interstitial fluid, why would it move out? Why would it go to the cells, right? Concentration gradient. It moves along concentration gradient because it's more in the capillaries, less in the interstitial fluid, so it goes from more to less. Now, plasma proteins. I mentioned that, look at these, let's say these are the pores of the capillaries, right? And look at the plasma proteins, it's like this. Can you fit plasma proteins within these pores? No. So plasma proteins that are inside the capillaries will stay inside the capillaries. There is no way that it can leave except if you have injury or inflammation or something wrong. But I'm talking about physiology now. I'm talking about normal. Normal, it's like this, and the pores are like this. There is no way to pass. What can pass through this? The basics of nutrients like glucose, it's tiny monosaccharide, water, gases, ions. These are all tiny, small H2O. This is small. But plasma proteins, like 200 amino acids together, that's too big. It usually doesn't move out. So which compartment contain more plasma proteins? Plasma or interstitial fluid? Huh? Plasma. More plasma proteins. So we said albumin, globulin, and fibrinogen. And the albumin, we talked about the transport function and osmotic. It's transport. Transport of what? Fatty acid, thyroid, and steroid. The three of them are hydrophobic. The three of them are lipid in nature. So meaning it carry lipid soluble things, anything that's lipid soluble. Globulin, antibodies. And that's why they call it immunoglobulins. So antibodies is the same as immunoglobulins. The globulins of the immune system. Okay, antibodies are the globulins of the immune system. So is it globulin? Yes. So this is the type of plasma protein, it's globulin. And it's also transport. Transport what? Proteins. Did you get the difference? Both albumin and globulin are transporters? Yes, but albumin mainly transport lipid soluble. And globulin mainly transport proteins. Okay? Any proteins in general. So if you forgot just proteins, like metalloproteins, hormone binding proteins, alloproteins, whatever the proteins are. Fibrinogen, it will change and become fibrin, which is important to stop bleeding, blood coagulation or blood clot. Serum. What's the difference between serum and plasma? Let's say they take a blood sample from you, okay? Take a blood sample. Let's say you're going to donate that blood. So they take it from you and they give it to somebody else, okay? It's not exposed to air, it's always kept in. You call this whole blood, right? Sometimes you want it to transfer to other people, but you don't need to give them blood cells. They don't have a problem with the blood cells. They only don't have enough plasma proteins, or their plasma is deficient. Yeah, sometimes it happens like this. Like some people, they have enough red blood cells, white blood cells, platelets. So if you give them, that will be extra. They don't need it. They need only the plasma part, the fluid part, for some reason, immunology region or something. Sometimes they do that. So what they're going to do is they separate those two. They leave it for some time. The blood cells will go down, sediment, and the upper part will be the plasma. So far, you still call it plasma, right? If you expose this to the air, the fibrinogen will be activated. It will become fibrin. Now we call it serum. This is the difference between serum and plasma. So what's the difference? Plasma is serum. But after the fibrinogen become fibrin, after changing the fibrinogen to fibrin, okay? Otherwise, it's the same. It was plasma, actually. Serum was plasma. Just the fibrinogen become fibrin. The other 1% is other things, hormones, enzymes, prohormones, and so on. Where do we make the plasma proteins? 90% of it, we make it in the liver. Like albumin, fibrinogen, we make that, most of it in the liver. How about globulin? The part of globulin, or the major part that's used for immunology, plasma cells make it. So plasma cells make antibodies, which is globulin. So where do we make our plasma proteins? Mainly in the liver. One exception, the antibodies, that is type of globulin, plasma cells. What are the plasma cells? Plasma cells. These are activated, what? B cells? Hello? Anybody? Plasma cells are activated B cells. Do you remember the B cells and T cells? Okay, that's great. Red blood cells is the major part, about 99%. So this is the major part, red blood cells, and we have some white blood cells and some platelets, but the major thing is red blood cells. Now, how to measure the amount of red blood cells? We always hear that, do you have a problem with your blood? Do you have anemia, for example? Do you have anemia? Let me count. Let me count the red blood cells. This is one way. It's not the only way. It's not just about the count, okay? So counting, how many do you have per microliter? So they take just small amount, they take a sample of the blood, and they put it under the microscope, they count and they multiply that several times, and they can tell you, you have this number, millions. This is one way. The other way is, and here's the reason. Look at this. This is a container, just random example numbers, right? This is a container that have 100 red cells, and each one is like this. It will be full, right? How about this? Another container, same size, 100, but like this. Isn't this is more than this? Did you get what I mean? No? Okay. What's the difference between filling this class with 200 people who are huge and 200 students who are tiny? Which 100 will fill the room? The 100 small ones will take half of the room, and the 100 that's huge will take the whole room, right? So is the size the same? It is not. So these large cells, normal cells, will carry more oxygen than this. So I cannot say how many RBCs do you have. If you have the normal number, you're fine. I have 5 million. Okay, you're fine. No, you're not fine. Because if this is 5 million of this type, that's too tiny, it will not be able to do the job. So how can we know now? Plus counting. There is another measurement that's called hematocrit. And the hematocrit is the packed cell volume. So they say, okay, this is the blood, let me know how much of it. Don't say it's 100. Don't say it's million. Don't say that. Tell me the percentage. Okay, so that's a better reflection of not the number, the amount. Because the amount is number and size, isn't it? The amount is not, it's a number and size, right? I have 100 liters and I have 100 gallons. Both of them are 100, but which one is bigger? 100 gallons, of course, right? So this is your other way, the second way, which is even more important than counting. It's hematocrit. So this is a packed RBCs. Let me know the percentage. This is your blood. Let me know the percentage between the cells and the fluid part, which is the plasma. They do a centrifuge. They put the blood in the centrifuge, which is something that rotates, spins really, really fast. And this is going to separate the components. The RBCs will be on one part, and the rest will be in other part. And tell me exactly, like this, I put it in this tube like this after centrifuge. Okay? So this is the whole thing. But this part is blood, and this is plasma. So you're going to tell me it's 50%, right? Look at this. Can you see it? This is a tube that contains blood, right? I put it in the centrifuge, so it rotates. Red blood cells are separated, and this is the cells, and this is the plasma. This is about 50-50, right? Like half is blood, red blood cells, and half is plasma. How about this? Maybe like 60% or something. This is the hematocrit, the percentage of the packed cells to the rest. And there are normal numbers that we will learn. Normal count, which one will be more? Males. It's just take it as a rule. This is like the basics of the blood. Anything in males is harder than females. Just take it like this. So count. 4.5 to 6.3 in males and 4 to 5.5 in females. Obviously, it's higher in males. How about the hematocrit? Males 4 to 52, females 3 to 47. Which one is higher? Obviously, males. Structure itself of the red blood cells, it's biconcave. So the middle part, which is this, is thinner, and the outer part is thicker, right? And the reason for that is to increase the surface area of the red blood cells. So there is a difference between this and this. Which one is longer? You think if you go around this versus this, this will have more surface, right? That's why the RBCs are like this. And actually, if that is lost for some reason, that will decrease the capacity of the RBCs to do the job. And these are abnormalities that we will talk about later. So the reason why the cells are like this is to increase the surface to volume ratio. They can go smoothly, they should go smoothly in the blood, moving around. They can actually bend. This is flexible. The structure that we were showing, it's actually flexible, so it can bend like this, okay? So it goes like this, and you can bend it, and then it will return back. So it's kind of flexible, so that it can go in blood vessels that sometimes is narrower, like this. This is a blood vessel. Okay, let's say the RBCs are here, and this is obviously larger than this, right? It can do like this. Squeeze itself. Can change the shape until it passes through, and then it will return. So it's flexible. The diameter is about 7.8 micro. Yeah, that's fine. Lifespan is about 120 days. Okay, you need to remember this. And what will happen to it after the 120 days? You break it down and recycle it. Can you leave the cells beyond that? You can't. It's useless. It can do the job for 120 days. This is a lifespan, 4 months. What's after the 4 months? It will not work. That's it. Lifespan is over. So what will happen after the 120 days? Send it to the liver and the spleen. They will break it down and use the components out of it, so you can replace it. So this is always turnover happening. Every 120 days, the red cells go to the spleen and liver, break it down, take the protein, use it again, take the iron and use it again, and get rid of the part that you don't need, okay? And you're gonna send it back to the bone marrow, and the bone marrow will replace it, and so on. Hemoglobin is a protein molecule. Hemoglobin. Okay. So hemoglobin, this is a protein molecule, and actually it's called hemoglobin. Hemo means blood. Globin is a protein. So this is a protein that's inside the red cells. Okay? And this is what can carry the oxygen. Oxygen and carbon dioxide, both of them. Hemoglobin, the normal, and this is a number that you need to remember, is 14 to 18. So there are a couple of numbers that you need to remember here. Okay? Hematocrit, the red blood count. Okay? The volume of the blood. Hemoglobin 14 to 18 gram per deciliter. This is adult male. The structure of the hemoglobin is quaternary structure, and I think you still remember primary, secondary, tertiary, and quaternary structure of protein. Do you remember amino acids? And then you start to fold, secondary structure, and then you put it in a whole structure, tertiary, and you have several units. That's a quaternary structure. So like this. Here's one tertiary, another tertiary, another tertiary, another tertiary. So 4 tertiaries together, that will make the quaternary, which is the final structure. So it's 4 subunits, and each one unit will contain 1 heme. Each 1 heme will contain 1 iron. Okay? The hemoglobin is 4 units, 4 subunits. Each one contains 1 heme. So how many hemes do you have? 4. Each heme will contain 1 iron. What's the importance of the iron? The iron is what attached to the oxygen. Okay? So oxygen, when it goes to the blood, it will attach itself to the iron. Okay? And now you call the hemoglobin, when the oxygen attached to the iron part of the hemoglobin, you call it oxyhemoglobin. What's oxyhemoglobin? It's hemoglobin with oxygen attached. Deoxyhemoglobin, on the other hand, is deoxy. So this is hemoglobin after you remove the oxygen. Fetal blood is a little bit different than adult blood because it has stronger affinity for oxygen. So you can say stealing the oxygen from the mom. Okay? Stealing the oxygen. They have better affinity, stronger affinity. So in the fetus, the fetal blood is trying to take oxygen, and the mom is trying to take the oxygen. So the fetus will be stronger affinity to take oxygen. Otherwise, it's far away from him. If the fetus doesn't have stronger affinity of the hemoglobin, he will not be able to get enough oxygen and he will not survive. So this is happening for a reason, to take the oxygen from mom. Carbaminohemoglobin, this is a term that means carbon dioxide attached to hemoglobin. So we have oxyhemoglobin, deoxyhemoglobin, carbaminohemoglobin. Oxyhemoglobin is hemoglobin with oxygen. Deoxyhemoglobin is hemoglobin after you take off oxygen. Carbaminohemoglobin is carbon dioxide attached. So basically the hemoglobin or the red blood cells carry oxygen and carbon dioxide, and it has different names. Anemia is when the hemoglobin or hematocrit levels go down. They used to, like up until 20 years ago or something, they used to say anemia is low red blood cell count. That's not true anymore. It's not true. I care more than the numbers. You should have normal numbers, but it's not only the numbers. It is the amount of hemoglobin that you actually have. Do you have 5 million and each one of them is like this? This is very low hemoglobin. If you have 4 million but it's like this is even larger, right? So it's not only the number. So when you define anemia, it's more of hematocrit or hemoglobin level, not just the number. Okay, this is the recycling of the RBCs. Here is the bone marrow. It makes red blood cells. Red blood cells do the function. 120 days later, part of it actually will be broken down anyway, and that's normal to occur. That will be hemolyzed and you get rid of it. This is something like, this is a loss, right? Like any factory or something, they always say, we're going to lose this amount, it's going away anyway. So 10% of it will be broken down and you get rid of it, and the 90% after 120 days, it will go to the macrophage of the spleen and liver. This is where it will be broken down. You break it down. Break it down into what? Protein and heme. Didn't we say hemoglobin? Right? Hemoglobin. So hemoglobin is heme and globin. Heme is what contains the iron. Globin is the amino acid. So when you break it down, you recycle the contents. How? You take the protein, which is amino acids, send it back to the bone marrow to use it. How about the heme? The heme contains very important structure, which is the iron. You cannot get rid of the iron. Also send it back to the bone marrow. So now the bone marrow have the amino acids, which is protein. It has the iron, so it's easy now to replace, to make others. How about the rest? The remnants of the heme, that will be changed to something called biliverdin. Biliverdin, so what's a biliverdin? This is the remnants of heme after removing the ferrous, or the iron, okay? That will change to bilirubin. Bilirubin go to the liver, and the liver will excrete it in the bile. Some of it will go excreted in the urine, and the rest will be excreted in the feces. So you excrete it basically, okay? One time real quick. Make the red blood cells, 10% broken down, 90%, 120 days. And go to the macrophage, break it down. Amino acids, go back, recycle. Ferrous, recycle. Ferrous part of the heme, recycle. So now you have the basic components. This is the most important part about the remnants of the heme. After you remove the ferrous, biliverdin, bilirubin, go to the liver, leave through the bile, go to the intestine. Some of it will be secreted in the urine, and other parts will be secreted in the stools. So this is how you recycle it. And you recycle about 3 million RBCs per second, so it's huge amount that's recycled all the time. You always recycle it. Hemoglobinuria and hematuria. Anytime you see terms like these, abnormalities like this, know that it's important. I'm not sure if I got these all together in the last slide or not. I don't remember right now, but most of the time I do that. So, but anytime you see something like that, know it's important. Okay? Hemoglobinuria, hemoglobin, urea. What does it mean? Huh? Hemoglobin, urea. Hemoglobin in the urine. Are we supposed to have hemoglobin in the urine? Of course not. Can we have the remnants like bilirubin? And yes, that's fine. These are just a little bit of remnants of the heme, just tiny parts that you're going to be excreted anyway. But the hemoglobin itself, no, you're not supposed to have it. And if you have it in very small amount, that can be an exception, but it shouldn't be there. And it happens if you have a lot of hemolysis. So if I see hemoglobin in your urine, I know that you're breaking down the blood. Instead of 10%, you're breaking down 20, 30%. So a lot of hemolysis going on for some reason. Hematuria, on the other hand, is not the same as hemoglobinuria, because hemoglobin is the main part of the red cells. It doesn't mean red cells, right? Red cells contain hemoglobin. So if the hemoglobin only appears in the urine, it means it's broken down. If the whole cell, you see actual red blood cells, this is called hematuria. So you got the difference now? And you don't, it have a totally different meaning. Hemoglobinuria, it means you broke down your RBCs, hemolysis happening for some reason. So the cells are actually opened, the hemoglobin inside is released, and it appears in the urine. So it means you're breaking down more than normal. This is something pathological. This is a problem here. But hematuria, it means you're leaking. Okay, so your blood vessels are broken down for some reason so that an entire RBCs or RBCs are leaving through, appearing in the urine. So we talked about the fate of hemoglobin, how it's going to be biliverdin, bilirubin, and so on. The iron itself will move from spot to spot. It will be called different names. When the iron is attached to protein, you call it transferrin. If you store it, you call it ferritin and hemostadrin. And you need to differentiate between those two. Okay, so iron during transport, you call it transferrin. If you store it, not transport it, just store it, leave it aside to be used later, that will be called ferritin. This is the main thing, and also hemostadrin. So bilirubin, biliverdin, and bilirubin is yellow, by the way. That come from breaking down the RBCs. I mean the heme part after you take the iron. The bilirubin is yellow, and this is the reason for jaundice. So what's the jaundice? The jaundice is build up of bilirubin. Are we supposed to have bilirubin? Yes, but you excrete it. So if something happened and start to build up, it will start to show up. Did you ever see somebody having jaundice before? Yellow eye and yellow skin. What is this? It's the bilirubin, which is, it's okay to be in the blood, small amounts until you get rid of it. But if you're not getting rid of it for some reason, like the liver is having a problem releasing it, or something wrong happened, or hemolysis is a lot beyond the capacity of excretion, it will start to build up and start to show up in different parts as yellow, which is called the jaundice. So it's supposed to be changed into urobilin and stercobilin. These are the forms that should be secreted. Otherwise, if it builds up in the blood, it will start to appear. Red blood cells maturation, you start with this, pro-erythroblast, and it will change to erythroblast. Just remove pro, and then polyhematocritic erythroblast, normoblast, until you get to reticulocytes. Most important of those is this in the beginning, those, and reticulocytes that will become mature cells. Why? Because if you are producing a lot of blood, you're losing blood. Let's say somebody's having bleeding and he don't know about it. You're having something bleeding inside. So you're losing blood and you're making blood all the time. Fast enough that you don't give the time to reticulocytes to become mature. You're rushing the bone marrow. I need more blood, more blood, more blood. You don't give it the time to maturate. So when you see reticulocytes, it means you have abnormal hemolysis. Erythropoiesis is the process of making erythrocytes itself, and hemocytoblast is the stem cells. And you need to remember this. Hemocytoblast. This is the mother cell, the stem cell. Okay, of what? Of everything, of all cells. This is a mother, this is a stem cell. And this will give you myeloid stem cells or lymphoid stem cells. Myeloid stem cells will do red blood cells and white blood cells. Lymphoid stem cells will do the exception, which is lymphocytes. So this is important to remember. And it goes like myeloid stem cell, proerythrocyte, erythroplasts, reticulocytes, mature RBCs. If you wanted to make, how to make red blood cells, you obviously need the amino acids, which is protein. You need the iron plus certain vitamins. It's not only amino acid and iron. You need vitamin B6, 12, and folic acid. Each one of those, if you don't have it, it will produce a different type of anemia. The most common and the one that we all know, iron deficiency anemia. Do you have anemia? Yes. So probably you're not getting enough iron, right? This is the most common one, but not the only one. Any one of these, if it is deficient, you will have anemia. You have to have all of these. So how about the malnourished people? Those people who are starving and they're not eating, they actually have anemia. Not because of the iron and vitamins, it's because of the amino acids. Iron is the most common, but also 6, 12, and folic acid will make other types of anemia. Pernicious anemia is the anemia that's due to vitamin B12 lack. So it's not only the iron. Not only the iron. You can have deficiency of vitamin B12. That will cause pernicious anemia. You have to remember this. Amino acids. Iron, 3 vitamins. If you are lacking one of the 5, you get anemia. Okay? 6, 12, folic acid. Any one of those deficient, B12, for example, is called the pernicious anemia. Erythropoietin is the hormone that stimulates, where is it secreted from, if you still remember? Erythropoietin, where is it secreted from? Kidney, yes, from the kidney. When not enough blood go to the kidney, it will secrete erythropoietin. Erythro means red blood cells. Poietin, making. This is going to stimulate the bone marrow to produce RBCs. So these are the different tests that we can do. We can do hematocrit, reticulocytic count. I just talked about this. Hemoglobin concentration. It should be between 12 and 4. RBCs count. So this is summarizing the numbers. And 1, 2, 3. These are the most important things. These numbers to remember. Okay, which is hematocrit, hemoglobin concentration, and the count. Surface antigen, the red blood cells will have an antigen like a protein on top, or glycoprotein on top, to tell the body what type of cell you are, to tell the immune system. So normally these antigens on the surface of the cells will be ignored. Your immune system should not attack your own cells. So the blood types are A, B, A and B and RH. These are the 3 different antigens, and you can mix them together different ways to tell what blood type you are. Do you all know your blood type? You should. You should know, just in case. And you should know both of them. Is it A? Is it B? Is it O? Is it AB? And is it positive or negative? So I'm AB positive. I'm O negative. Okay, I am B positive. Okay, so AB is one, is this type of antigen, and the RH is another thing. Okay, these are different antigen that can differentiate between the cells. And type A, basically, just remember this and you will get it. The name comes from the antigen. the name of the group come from the antigen. So type A contain antigen A. Type B contain antigen B. Type AB contain both of them. Type O, and O by the way means 0. O means 0. It doesn't have any antigen. Can you remember it this way? Okay. So think about this. This is the lesson. If you have antigen A, what should your body produce? What type of antibody?
B.
A or B?
B.
B, of course. You cannot produce A. It will fight your own cells, right? So if you wanted to get transfusion, what type you get? That contain what? Contain anti-B or anti-A? Anti-B is okay. It's okay with you, right? What's the anti-B? It's your own type or O. Okay? Because O doesn't have any antigens. You have your anti-B, so I cannot give you B, right? You have anti-B that's ready and waiting for the B. Once you give me B, I will attack it. Don't give B. How about O? Yeah, it doesn't have anything. Not O, not B, so that's okay. How about A itself? That's fine, and so on, okay? So if you have an antigen, you cannot have the antibody against that same antigen. So AB, for example, what's the common name for AB? Like my blood type is AB. What do you call it? Universal? Universal what? Donor is O. You can give O to anybody. How about AB? Universal acceptor. It can take from anybody. So I can accept blood from anybody. Can accept from A, B, O, anything. So this is called acceptor. Why? You don't have any antibodies. So what are you afraid of, right? You gave me A, I don't have antibodies. You gave me B, I don't have antibodies. You gave me O, it doesn't have antibodies in the first place, right? And that's it for today.